Have you ever really thought about how a tiny paper cut just heals itself? Or how a massive tree can grow from a single tiny seed? Well, today, we're diving into the incredible biological script that makes all of that possible. We're talking about the cell cycle.
Seriously, think about it for a second. That little cut you got last week, you watch it day by day, and it just magically disappears. How does that happen? How did you happen? You started as a single cell, and now look at you.
The answer to all of it is this amazing microscopic drama that's playing out millions, probably billions of times a second inside your body. Right now, it's this incredibly precise process, a fundamental dance of life itself.
And that process is called the cell cycle. Now, okay, that definition sounds super technical, I get it, but really just think of it as a cell's entire life story. It's this perfectly planned out performance, from the moment it's born to the second it divides and creates two new identical cells. It's all about making a perfect copy of its DNA and all its other parts for the next generation.
And this, right here, is our guide for the journey. This is the visual roadmap of a cell's life. We're going to follow this thing step by step, from its birth all the way to the big split. So, let's get into it.
So, this whole story unfolds in two main acts. First up, you've got interphase. This is the long, careful, get everything ready phase. Then you have the M phase, which is the short, super dramatic finale where the cell actually divides. So it's all about prep, and then the main event.
And what's really wild is how much time the cell spends in each act. I mean, look at this. The prep work, interphase, is a total marathon. It takes up almost the entire life of the cell. The actual division, that's just a quick sprint at the very end.
Yeah, interphase takes up a mind-blowing 95% of the cycle. To put that in perspective, for a typical human cell that divides every 24 hours, it spends about 23 of those hours just getting ready. Everything, all the critical groundwork to make sure the division is perfect, happens during this long prep phase.
So what in the world is going on during that 23-hour marathon? Let's zoom in on Act 1, interphase, because it's not just the cells sitting around waiting. No way. It's a super busy period with three distinct stages. Okay, following our roadmap, the first stop is the G1 phase, or gap 1. You can think of this as the cell's day job. It's doing what it's supposed to do, being a muscle cell or a skin cell or whatever, and all the while it's growing bigger.
Next up, the cell begins its most important mission, the S phase. The S stands for synthesis. This is where the cell has to make a perfect, flawless copy of its entire library of DNA. This is absolutely crucial, because each new cell needs a complete set of instructions.
To keep track of this, let's use a little shorthand. We'll say the starting amount of DNA in the G1 phase is 2C. Just think of it as one complete set of genetic info. That's our baseline.
And then, after the S phase is over, something amazing has happened. Every single chromosome has been duplicated, so now the cell has double the amount of DNA. It went from our baseline of 2C all the way to 4C. Now, this is a super important point, and it can be a little confusing, so stick with me. Even though the amount of DNA has doubled to 4C, the actual number of chromosomes is still the same. So if the cell started with two sets of chromosomes, what we call diploid, or 2N, it's still 2N. It's just that now each chromosome is made of two identical sister copies, kind of stuck together. Think of it like a genetic 2-for-1 deal.
Okay, DNA is copied. Now the cell moves into the G2 phase, or gap 2. This is like the final dress rehearsal before the big show. The cell makes any last-minute proteins it needs, grows a little more, and does one final check to make sure everything is absolutely perfect for division.
But wait, what about cells that aren't dividing all the time? I mean, not every cell in your body is constantly going through this cycle. Turns out, there's a built-in exit ramp for cells that need to take a break or maybe even retire from dividing altogether.
This off-ramp leads to a state we call the quiescent stage, or G0. Cells in G0 have basically just pulled off the highway from the G1 phase. They're not asleep or anything, they're still working hard, doing their specialized jobs. They're just chilling, waiting for a signal to get back in the fast lane if they're needed.
So, which cells are taking this exit? Well, a lot of the really specialized ones, like your heart cells and your nerve cells. Others are just in G0 temporarily, kind of on standby. They're ready to jump back into the cycle when you need them, like when you get a cut and need to make new skin cells, fast.
All right, our cell has done all its prep work, it avoided that G0 exit ramp, and now it's showtime.
It's time for the main event, the grand finale, the M phase. This is where all that preparation finally pays off.
The M phase is basically a two-step dance. First, you have karyokinesis. That's the fancy word for the nucleus dividing, and all those duplicated chromosomes getting pulled apart to opposite sides of the cell. Then, right after that, you get cytokinesis, which is when the cytoplasm splits right down the middle, creating two separate cells.
And there you have it. The result is two brand new daughter cells, and each one is a perfect genetic clone of the cell that started it all, ready to go on its own journey through the cell cycle. And so the whole thing begins again.
Wow, that was a whirlwind tour of a cell's life, right? So to help all this information stick, let's pull it all together with a quick and easy cheat sheet for the whole cycle. This is where we lock it all in.
Here you go. This table is your at-a-glance guide. Remember G1? That's cell growth, and our DNA content is 2C. Then the super important S phase, where DNA replication doubles that content to 4C. In G2, the cell does its final prep, still at 4C. And finally, in the M phase, division happens, and the DNA content in the two new cells goes right back to 2C. And don't forget the G0 exit, where resting cells are also at 2C.
So if you remember anything, let it be this. For a typical human cell, the chromosome number, 2N, stays the same all through interphase. The DNA content starts at 2C, doubles to 4C, and then goes back to 2C after division. And the whole show for a human cell takes about 24 hours. Got it? Awesome.
From healing a cut to our own growth from a single cell, the cell cycle is just a masterpiece of biological precision. But that brings up a really big question, doesn't it? What happens when this perfectly controlled system breaks down? What happens when cells just start dividing, and they just don't stop? Well, that is a whole other story.